So I'll speak in English because we have a school going on where some people don't speak Portuguese. So Eduardo couldn't be here. This is the time of our colloquium. So I will do the presentation, but this is just for people who don't know. This is a colloquium. It's a part of the school. We have a school going on. And it's also a special outreach program we have about distinguished lecturers. So for people who are not part of the school or a part of the IFT, you're probably here for that reason. So if you want, Anna is organizing our outreach activities, and if you want, you can, if you haven't signed up your name to get emails about other lectures, we'll have outreach lectures. So you can talk to her after the colloquium. Okay, so it's a pleasure to have Curtis Callan here. So he's been to Brazil a few times already, but I think this is the first time he's been in São Paulo.
That is correct.
So for people who don't know him, he's well known to the high energy physics community for the work he did on QCD and strong interactions and string theory. And he won various medals, Sakurai Prize, Dirac Medal. He's been the president of the American Physical Society. I'm not sure that's a prize or, but, sorry? Okay, I can speak louder. So I'm not going to repeat. He's got various medals, and he's been a professor at Princeton now since 1970-something. Or
72 to be precise, yeah.
So, but the reason why he's here at this school is because for the last 12 or 13 years, he's been working in problems related to biology, which I think is going to be a common aspect of many physicists in the future, and he's going to tell us here in the colloquium why, I guess.
Okay. Thank you, Nathan. It's a pleasure to be here. So I'm here for the School on Mathematical Biology and attempting to say things which are quite, how shall I put it, analytical and detailed and too complicated to talk about in a general colloquium. And Nathan asked me if I would give a rerun of a talk that I gave a couple of years ago at a similar institution, at the Kavli Institute for Theoretical Physics in Santa Barbara, and it was an outreach lecture there too, and the talk was meant to be for the general public, but sort of general public with a level of comfort with the kind of reasoning that physicists like to make involving equations and quantitative ways of addressing phenomena was a little bit higher than that for the average public. So bear with me. I'll try to not push my luck too far. So the title is meant to be provocative, The Biological Future of Theoretical Physics. And I have an abstract which still seems to work. It says the very success of theoretical physics in elucidating the structure of matter at the smallest scales and of the universe at the largest scales has made the future course of the discipline uncertain. At the same time, the new ability of biological experiment to produce massive data is creating an urgent need for mathematical frameworks in biology, frameworks of the kind that theoretical physics has traditionally provided for physical science. So I think you can say that there are overlapping crises, and I would like to argue that they offer a golden opportunity for both disciplines to collaborate. And this kind of collaboration is what's happening in this school, so this is a perfectly appropriate place to make this argument. I'll expand on the theme and sketch some specific examples of how theoretical physicists are taking up the challenge. I have to be sketchy on that last part, because that descends to the level of equations which are not easy to grasp, in any concrete way. But I'll sprinkle some of them here and there, but hopefully not too many. Okay, so Nathan gave a brief statement of who I am and why I'm here. I'd like to go into that a little bit more deeply, briefly. I used to be a theoretical particle physicist, okay? And I wrote papers that had, well, more recently, things that refer back to what Nathan mentioned as string theory. And about 10 years ago, I started writing papers that had completely different sounding titles. Precise physical models of protein-DNA interaction from high throughput data, information capacity of genetic regulatory elements. I'm occasionally asked, why did I do this? Why did I switch, in quotation marks, to biology? Well, not really because of any dissatisfaction with mainstream theoretical physics. It was rather that I had some colleagues who were visionary and pioneering who showed me that biology poses fascinating questions for theory, that you could, somebody trained as a theoretical physicist to like to construct mathematical frameworks to deal with the real world could enjoy doing, and that the time is ripe to attack them. Now, thinking about this a little bit further, it seems to me that biology and theoretical physics have both come to a crisis that offers opportunities for both subjects. And my purpose here is to explain what I mean by this. By the way, at the top, I say, with apologies to Admiral Stockdale, only Americans of a certain age can remember what that joke refers to. There was I guess it was in 1988, the vice presidential debate, during the presidential campaign was between Senator Lloyd Benson and somebody that nobody had ever heard of, Admiral Stockdale. He was the Republican vice presidential, he was the vice presidential candidate. And he opened his statements by saying, "Who am I and why am I here?" And this was definitely a useful thing for him, and I think maybe it's a useful thing for me to say. Okay, so theoretical physics, for those of you who practice it, and those of you who read about it in the scientific literature, and those of you who follow it in the, semi-popular press, is a hugely ambitious enterprise. Okay, it describes how our universe came into being from the Big Bang. It follows it through 13.7 billion years of expansion, during which time the universe that we see when we look out at the universe with telescopes or cosmic microwave background detecting telescopes came together. Gas clouds formed, the gas clouds collapsed due to fluctuations in density into things that eventually became galaxies and stars and black holes, we now know. They are observed with the gravitational wave detecting devices. And basically, the laws of physics that we've put together over the last couple of centuries really do describe the behavior of inanimate matter absolutely everywhere in our universe back to, fractions of a second after the Big Bang singularity. Quite remarkable. It comprehends a lot, but some things escape its net at the moment. And this is a little cartoon of the stuff that is not caught in the net yet of theoretical physics. The emergence of living organisms that become more and more complex and, by some measure, more and more intelligent, although one can always have one's doubts about the evolution of intelligence past a certain point. Okay, so what's the primary task of theoretical physics? Well, I get into trouble when I say this, but I would say that, at least, I grew up to believe that what theoretical physics was in the first instance about was to discover the fundamental mathematically expressed laws of nature, and, of course, to identify the stuff, right, the matter or related entities that obey those laws in our universe. All right? Now, we are very, very lucky. These laws are valid within broad domains of phenomena and are, in some sense, simple. If you are willing to use the right kind of shorthand, most of them can be written on a 3x5 card. So it's not too hard to remember Maxwell's equations or Newton's equations or even the equations of general relativity. Quantum mechanics, at least in the form of the Schrödinger equation for atoms, can be written down quite succinctly. This is, of course, a data compression of massive quantities. You have to spend years learning what these shorthands actually mean and how you can turn them into useful predictions. These laws were discovered in steps over recent centuries. We all know about Newtonian mechanics and the gravitational force law in the 17th century, Maxwell's equations and special relativity in the latter half of the 19th century, Einstein's general relativity in the early part of the 20th, quantum mechanics of electrons, atoms, and molecules in the early 20th century, quantum field theory, the standard model of everything, 1970s, Big Bang cosmology and a theory of the origin of our universe. That overlaps actually with recent history. Anyway, the culmination of this development is the outcome of an explosion of discovery over the last 50 years, and that is typically referred to as the standard model of our world. I'll be a little bit more precise about what is in the standard model in a second. It's a precise mathematical theory whose scope is, as I said, pretty damn much everything, right? We start fractions of a second after the Big Bang, and we continue on until now, and capture, in principle, everything in the inanimate universe. Some modesty, of course, is in order, but I think the human race is to be congratulated on getting this far. Okay, so what is this standard model consensus? Some people don't like the term standard model. I don't know how it came to be the accepted term for our current, in some sense, nearly ultimate theoretical synthesis, but this is what it is. It could be called the standard theory, and that would be equally good. The interesting thing is that 50 years ago, when I was a graduate student, there was no agreement on the fundamental nature of matter, no agreement on the physical theory governing that matter, or how the universe worked at the larger scales. And over time, within my lifetime, a picture and theory of matter in the universe came together. And I think there are two tracks. One was the particle physics track, namely, eventually, experiment and theoretical thrashing about identified a specific quantum field theory of the strong, weak, and electromagnetic interactions between 3 generations of point-light particles called quarks and leptons, forces between these things mediated by gauge gluons, and these are things that are like, in many ways, the photon that's responsible for phenomena of electromagnetism, and the W and Z bosons, which are massive versions of the photon, and the famous Higgs boson, just actually physically grasped, if you like, just a few years ago at the accelerator at CERN. This whole complex of equations and entities which obey the equations requires a certain number of parameters to tell you exactly what's going to happen if you set the machine going. You have to specify, well, what's the mass of the electron? What is the strength of the electromagnetic coupling constant? There are a bunch of other masses of the quarks and leptons, and so on and so on. But if you add them all up, the number of parameters needed, in a certain sense, to completely specify this theoretical framework is rather modest. Something of the order of 21 parameters. Perhaps you can count them slightly differently. The theory so constructed manifests, as far as we can now tell, with just 1 or 2 tiny disagreements, total agreement with many and varied experiments. One of the continuing threads of discovery in fundamental physics or particle physics was looking for things within inside things. The human race identified atoms as a fundamental constituent, sort of philosophically at the time of the Greeks, but realistically in the terms of chemistry at the end of the 18th and beginning of the 19th century. And much of what happened after that was to discover, these 92 different atoms out of which one constructs the chemical world, or the material world, are themselves made out of stuff, electrons and nuclei. The nuclei are made out of a few constituents, neutrons and protons. The neutrons and protons are made out of a few constituents, quarks and gluons. Now, you could assign a size to the atom, some small number of angstroms in diameter. You could assign a size to the neutron, some 10 to the minus 4 times less than an angstrom. You can't assign a size to the electron or to the quarks. These are, at some fundamental level, they are point particles in the sense of Euclid. They have no inside. You can't say that there's some structure that could be in various different ways, so that you would need lots and lots of numbers to specify what it is. The fact that they're point particles means there's nothing inside them, and the number of qualities that they can have is very limited. They can have a mass, they can have various kinds of charge, and they can have spin, and that's it. It's a finite set of numbers. So there's a certain sense in which, at the stage we are now, the search for constituents, which was in some sense the unpeeling of the onion, the peeling of the onion story of particle physics for many years, has gotten to a point where it's, in some sense, it's stopped. You've gotten as far as you can go. I'll elaborate on that in a bit. There's also the cosmology track, right? The recession of the galaxies, which was observed by doing spectroscopy of galaxies and stars back in the early 20th century, eventually led to the conclusion that if you look out into the universe, the objects that are further and further away are receding from us faster and faster. And this recession suggests that the whole thing basically was a lump of matter that exploded at some point, and the stuff that emerged from the explosion going faster after a given time got further away. That's why we see this, so to say, separation of objects on the basis of distance and velocity correlated with one another. The more interesting thing is that if things really started in a high temperature, high density initial state, which is this recession of the galaxies inevitably suggests, then there should be a thermal afterglow. It was very hot. If something's hot, it radiates. If it radiates, you can look at the frequency distribution of the radiation and determine the temperature. Well, if you follow that line of argument, you conclude that out of the Big Bang, there emerged at one point free-flowing, very hot thermal radiation, and as the universe expanded, basically, this radiation adiabatically cooled, the photons got longer and longer wavelength, and doing back-of-the-envelope calculations that were completely right but nobody believed for a long time, you would conclude that now, if you look out into the universe, it should be as if you were looking into an oven at a few degrees K, a few degrees Kelvin, close to absolute zero, but quite measurably different from absolute zero. And if you had the right kind of apparatus, you should be able to detect this radiation. And then from the properties of the radiation, you could begin to infer details about what happened at that initial instant. And that of course, came to pass. We now measure the thermal blackbody radiation that was released by the Big Bang in great detail. And two surprises came out of this analysis. The matter of particle physics, the electrons, the quarks, the neutrons, the protons, and whatnot, are there in the energy balance of the universe, but it is a minority player. Both dark matter and dark energy dominate and seem to act on the world only via gravity. This explains many puzzles of astronomy and also how today's universe coalesced from the Big Bang. So two very remarkable threads of discovery that went by steps over the last 50, 60, 70 years combined to give you a very detailed description of how the universe, how the material universe acts and evolved over a rather long period of time, the 13.7 billion years from the Big Bang. So let's go a little bit deeper into what is the standard model of particle physics. Well, I said basically it is a list of entities that interact with one another in a way that is specified by a certain type of quantum field theory. If you don't study quantum field theory, you probably don't have a concrete notion of what it is. It's basically a way of dealing with the inevitable capacity of fluctuations in the vacuum to create particles by pairs of particle and antiparticle, and how to turn that into a quantitative dynamical theory. So the things that can be created and destroyed in the quantum field theory that we have to work with because it describes the world we're in, has a finite number of components, and they're organized in, they're helpfully organized, I hope, in this matrix way. There are the things out of which most of the matter we see are stably made. The up and the down, these are called quarks. These guys in this rectangle up here, they're called quarks. They are things that are in many ways like the electron. They're spin 1/2. They're, as I said, point particle constituents. In addition to having electric charge, they also, however, have the ability to interact with things called the gluon, which are an analog of the photon that essentially causes these quark objects to interact with one another. These guys are like the electron in the sense that they, not only do they have spin and charges, but they have mass. And the first group of up quark, down quark, electron, and neutrino, I'll say a word about a neutrino in a second. This is called the first generation. There's a somewhat heavier generation of objects. There's a third, much more heavy generation of objects that are they are carbon copies of their predecessors, except for a few irrelevant parameters like what's their mass? You know, how much do they weigh if you weigh them? How much do they resist acceleration if you put forces on them? Then there are these things that are analogs of the photon. The photon is the particle of light, and it's a boson. It's a spin 1 particle. The photon itself carries no charge, but it is created by charge. Its fellows, like the gluon or the Z and the W boson, which are responsible for other classes of interactions, do carry charges. Some of them do, and have mass. The photon is the unique guy in the whole game that has no mass at all. And the Higgs boson is a kind of outlier, but you couldn't have this whole game without it. It's a scalar particle. It has no intrinsic spin. It has a mass, and it is responsible for the spontaneous breaking of symmetries that otherwise would have made all of these particles massless. It's because this guy exists and has a non-zero mass that these guys have mass. And there's an intricate structure which ties all of this together in a set of equations which, if you're a sufficiently well-trained specialist, you can work with, and you can calculate decay rates and cross sections for interactions to occur and all the rest of that. It's a whole art form. All right, so we've got a very specific quantum field theory of how these point particle constituents interact. The various numbers that govern their behavior, their masses, and their coupling constants are known. One rather interesting feature is that these things that are constituents of the strongly interacting particles, like the neutron and the proton, they have a peculiar property called confinement. That is to say, we know that the proton is made by putting together 2 up quarks and 1 down quark. And these guys are glued together, and they're very stably bound to each other. You might think that you would be able, by bashing the proton hard enough, to drive one of its constituent spin 1/2 quarks out to become a free particle that you can observe directly. One of the amusing features of these guys is that if the unit of charge is defined by the electron, these guys have fractional charge. So if you were able to blast a constituent quark out of a proton or a neutron, it would have a rather definitive signature. It would carry charge 1/3 or charge 2/3, and that would be something that you would be able to see. One of the remarkable features of the theory that holds all of this together is that all evidence points to there rigorously being a property called confinement, which is that if you take a collection of quarks that are, so to say, bound together in a stable state, if you attempt to pull it apart into its constituents, you can't do it. You can never extract a free quark. We're not able to prove this, but our confidence that this is a true property of the theory is very high for reasons I can't go into now. Our ability to calculate specific results in this theory, somehow I missed a proofreading step, is limited, but it's good enough to make us strongly convinced of its accuracy. You have to remember that we all believe in the Schrödinger quantum mechanical theory of atoms and molecules, but we can actually analytically calculate the predictions of the Schrödinger theory for atoms or molecules only in a very small number of highly limited cases. We are convinced from all kinds of other evidence, better and better approximate calculations, et cetera, that involve more complex atoms than, say, the hydrogen atom, that yes, this quantum theory is not just a good theory, it's an exact theory. And the same is true of the Standard Model, which takes the whole thing to a higher level. Okay, let's talk about cosmology for the moment. At the same time that this theoretical model of particle physics, the constituents and forces, was coming together, observation and theory was giving a more and more precise picture of what the universe looks like in the large. So this is a timeline down here, starting at 0 seconds, 1 second, 1,000,000 seconds, 10 to the 12 seconds, 10 to the 18th seconds. 10 to the 18 seconds is roughly now, after the Big Bang. And I'm following a variety of things that happened as you marched along. Now, so you start out at the very beginning of the Big Bang in a situation of extremely high density, high energy, high temperature. As the expansion goes forward, the temperature of this mess decreases, and various things happen. If you're willing to wait until 300,000 years after the Big Bang, the temperature has gotten down to the point where it's still mighty hot. Atoms are ionized, but they're just about on the point of becoming unionized. The temperature is just about to fall below that at which atoms are preferentially ionized. The electrons, at least the outer electrons, have been driven off. When that happens, when you pass from ionized gas to unionized gas, the photons that are hanging about, that at that point are imprinted with whatever is imprinted on them in the way of distributions of energies by the temperature of whatever temperature roughly 1 eV corresponds to. Past that point, as the universe expands further and cools further, the photons, as they say, free stream. They don't interact with each other. They no longer are modified by the matter that's out there. They simply free stream. And it's those photons that got released at this moment when the universe got cool enough so that atoms were no longer ionized. And that happened at about 300,000 years after the beginning. These photons just free stream. And when we look up and observe the cosmic microwave background radiation, we're seeing those photons. And we're seeing, and if we look at their distribution in energy, we can, in effect, infer what was the temperature when they were released. Or, put it another way, what is, if those photons were put into a box right now and just observed as black body radiation, what temperature would you give them? Well, the average answer is 2.75 degrees Kelvin. And if you look across the sky and plot, if I look in this direction, what is the temperature of the black body radiation? If I look in that direction, what is it? If I look in that direction, what is it? On the average, it's 2.75 degrees Kelvin, but there are small variations of approximately 1 part in a million. And satellite experiments have been done over the decades. Balloon-borne experiments, satellite experiments have been done over time that have allowed us to make a map of what is the temperature of the black body of the cosmic microwave background sky. And the little red dots are places where it's a few millionths of a degree more than average, and the blue places are where it's a few millionths of a degree less than average, and so on and so on. It turns out that this inhomogeneity, very, very tiny inhomogeneity, but inhomogeneity nonetheless, is why we're here. Because this means that there's a variation in the energy density. There was a variation in the energy density of the universe from place to place that was reflected in the variation of the temperature that we observe. And if you have a gas that has a little bit more matter density here than here, if you let it hang around for long enough, gravity is going to exacerbate that. The places where there's more mass is going to accumulate by gravitational attraction more mass, stealing it from the place where there is less mass. So if you can turn this into a map of what's the matter density, and then propagate it forward in time just using good old Newtonian mechanics, you will soon see that these little tiny ripples are going to turn into really humongous accumulations of gas with empty space between them. And those humongous accumulations of gas are going to eventually start causing within themselves further concentrations that are going to turn into stars. And so, roughly speaking, 500 million years after the Big Bang, the collapsing of the inhomogeneities in matter density in the universe brought stars and galaxies to life. And you can follow that quite rigorously, just integrating forward or integrating backward using the equations of Newtonian mechanics, with a little bit of general relativity thrown in, because this is in an expanding universe. Now, there's a part of this that's really quite interesting. The way in which the universe expands is governed by pressure and temperature, and the pressure and temperature feedback. It depends on the distribution of energy and pressure in the matter inside the universe, and by an elaborate sequence of arguments that involve only physics that we really understand, we can, from observing the way these inhomogeneities are distributed in size and in space, we can infer a lot about the nature of the matter that was present at the moment that these inhomogeneities were formed. You find the following thing. At the moment of decoupling, 300,000 years after the Big Bang, you can do a census of what were the various constituents that were responsible for the mass and energy of the universe at that time. And roughly speaking, you discover that atoms, the stuff that everything around here is made of, were responsible only for 12% of the energy density. Photons, the stuff that was eventually going to free stream and become the microwave background, was responsible for 15%. Neutrinos, they're partners of the electron in the Standard Model. They're very, very light. We used to think they were zero mass, but I think now we have very strong evidence that they have rather tiny masses, but non-zero. But then there's a whole other chunk called dark matter. Dark matter is stuff that's out there in particulate form, probably, but we don't know what it is. It has the energy momentum tensor properties of ordinary dust or atoms, if you like, but it doesn't have any pressure. It doesn't interact with itself. So there's another component called dark matter whose presence is inescapable, and the relative importance of dark matter and the matter that we know, the matter that can light up when it gets hot and turns into a star, the relative proportions of these things at different epochs in the universe varies. In fact, today, we know that atoms make up roughly 5% of the total energy budget. Dark matter, roughly, well, it says 23, I suppose that's roughly right. And the rest is something even crazier called dark energy, which is, essentially it's what's called the cosmological constant. That is to say, you can attribute an energy and a pressure to empty space. And apparently, you have to, in order to properly account for the dynamics that we see following these things as they expand forward in time. So I'm not going to go deeply into dark energy or dark matter today. On the other hand, this should be taken as a very, very strong caveat to what I said before about the extent to which we understand what's going on in the universe, because we have discovered that there's a very significant component of material energy in the universe that we actually don't have a clue about. We have various ways of looking for it, various hypotheses about what it might be, but it's clearly outside the Standard Model. So there's something there. You can get away with, you can understand a very large fraction of what's going on in the universe without it, but it remains a major mystery. And then there is dark energy, which I will not talk about very much. And okay, so let me take this summary as the occasion for making a remark on how science is done these days. All right, so in cosmology and particle physics, we actually don't directly observe the things we're really interested in. All right? We don't pick a quark out of a nucleon and put it over on a scale and measure its mass. We have to look for its properties by indirection. And a particularly good example of this comes from the Big Bang story, the cosmic microwave background radiation. So the idea is, well, so for example, the experimental evidence that we have about the cosmic microwave background is this map of temperature on the sky, okay? So it's a few million data points in the most recent data release from Planck. And so this is hot spots and cold spots on the sky as a function of where you are on the stellar sphere, function of where you are in angle. So what people do with this is to turn it into a correlation function. That is to say, you'll notice that the places where it's cold tend to stick together. The places where it's hot tend to stick together on the sky. In order to get a temperature that's very different from the local temperature, you have to move a certain distance on the sky. So there's something called the angular correlation function, which captures the way in which there's a persistence in angle of the fluctuations in temperature. So if you perform a certain Fourier transform of this data set, you get a so-called power spectrum of the temperature fluctuations as a function of angular size. And what you see is the biggest peak is at some angular distribution, at some angular size, which is a little bit less than half a degree, and there are wiggles, all right? That's what you can get directly from the data. The correlation of temperature variation with temperature variation somewhere else varies as a function of angular separation, and if you plot the function that you get when you construct this correlation function, it has these wiggles. All right. I can't give you the whole story, but it is the existence of these wiggles, how big they are and where they are in angle, that allow one to make this conclusion that's represented by this pie chart down here, where you infer what's the proportion of the matter in the universe at that time, which is dark matter, what proportion neutrinos, what proportion photons, what proportion atoms. This is a very elaborate analysis, and basically it has to do with it has among other things to do with, well, what's the propagation speed of sound in a medium that properly describes what's going on at this time. That's what governs how far away the influence of a particular fluctuation in temperature can get in the time that's available to it since the beginning of the Big Bang. So there's an underlying model of what's going on, and this model is very simple. It says the matter of the universe is composed of certain components. I know something about the dynamics of the expansion of the universe, and I attempt to find a combination of parameters. What fraction of the universe is made up of which of these components at this time to match this data. So I have very noisy data, lots and lots of fluctuating temperature values. I can do some simple data analysis on it, but that doesn't directly tell me anything. I have to have some picture of what's going on that can be turned into a set of equations that reflect how things must have been happening at that time, given what we know about Newtonian mechanics, general relativity, and so on, and adjust the parameters, the rather small number of parameters that I would need to fully characterize the model of the universe at this time, to find the ones that fit. All right, now, of course, the red line here is the theoretical prediction. The error bars are the actual data error bars. So you can see that having found the best values of the parameters that described the universe back then, the match to the data is really quite extraordinary. Now, all I want to say is that what we've done here is we have a noisy data set. We have an underlying simple model for how the things that you're interested in, would affect the noisy data sets that we do measure, and we use statistical inference to see through the noise to find the values of the underlying parameters. This is actually a nearly universal method in modern physical science. And one of the things that I would like to argue, maybe get across, is that biology is entering an era in which it will need to do the same thing. In cosmology and particle physics, we are fully into this and have been into this for decades, but biology is getting there too. Okay, now, to get where we are, massive resources were deployed. We all know about, I'm sure you've heard about the Large Hadron Collider at CERN. This is an aerial view of Geneva, Switzerland to the, I guess it's to the west of the airport. There's the runway of the Geneva airport. The circle here represents the tunnel in which protons are accelerated to 13 point whatever it is, giga electron volts. They are collided against protons running in the opposite direction at a few specific interaction points, and the debris from these collisions are then detected and quantified and analyzed, well, quantified in detectors like this guy right here. This is the compact muon solenoid detector. It's not very compact. These are human beings, and this is the detector itself, and the particle beam comes through here, and collisions take place in the center of the sky, and all of this stuff is electronics which track the charged particles that come flying out of the collisions. So what you get as data is showers of charged particles emerging from collisions. You measure the energies and directions of propagation of these charged particles. And from that, with the help of the underlying theoretical model, you eventually extract things like the mass of the Higgs boson is 125 GeV, et cetera. All right? And similarly for the cosmology experiments, they don't look as immense. This is, roughly speaking, a $10 billion enterprise. The satellites that were used to measure in great detail the cosmic microwave background are kind of billion-dollar enterprises. It's a major societal effort to acquire the data that eventually we were able to make sense of in the way I've described. Okay, so this is, we have engaged in a sustained effort to answer what amounts to a question of natural philosophy. We're not curing any disease by discovering that there was a Big Bang 13.7 billion years ago, nor are we curing disease or making people richer by discovering that the Higgs boson exists and has a certain mass. Although it is worth noting that the internet is in some sense the practical utility, the commercial utility of the internet at some level grew out of innovations that were created at CERN or by people at CERN who were trying to facilitate the collaboration of the thousands of scientists who are needed in order to make this whole enterprise of a high, very, very high level of technology work, and then also to make sense of the data that comes out of it. Okay, so massive resources were deployed. Where does theoretical physics go from here? Well, I'll just say a few words. There are loose ends, but the historic program has succeeded so well that you could argue that it's put its own continuation into some serious question. So on the conceptual side, we're in an interesting position. You know, once you're down to point particle constituents of matter, you can't really explain them in terms of more fundamental entities. At some level, that part of the game is over. Now, there could be more massive point constituents that even the LHC can't see. And as I say, neutrino masses and dark matter point that way in an obscure fashion that we haven't been able to penetrate. Going deeper in the light of string theory, we can see a natural reductionist limit point of the Planck scale, which is way beyond direct experimental reach. On the experimental side, exploring relevant energy scales is increasingly costly and surely approaching societal limits. And in the long run, okay, so it's going to be hard to do the kinds of experiments that one can imagine doing in order to address these hanging questions about our understanding of fundamental theory. So life is going to be hard for fundamental theory from here on. Not impossible, to be sure, but I think it's worth asking whether there are other paths that one might take in parallel, not instead of. Okay, now there is an other agenda of theoretical physics. And so I said, my take when I went into physics long ago was that the point was to discover the fundamental laws. Well, indeed, discovering the fundamental laws is the historic core mission, but it's not all that theoretical physicists do. In addition, you want to explain phenomena that are not directly baked into the fundamental laws. In other words, to be able to understand and ideally to predict emergent phenomena. So you perhaps have heard emergent phenomena as a buzzword in the past, but let me just mention a couple of instances to show what I mean. So for example, superconductivity is a rather striking, really first order phenomenon. If you cool certain metals below a certain a critical temperature, they lose all electrical resistance. I mean, it's literally zero. It's not small, it's zero. And to show that superconductivity follows from the Schrödinger equation for electrons moving in a host atomic lattice, that is a gigantic challenge, okay? That is what so superconductivity is a phenomenon that, you know, once you see it experimentally, you know it. It's very, very different. It's qualitatively different from anything else. The Schrödinger equation, as you write it down, doesn't say anything about superconductivity. It says, well, I can calculate the energy levels of atoms containing so and so many electrons, or if I put a bunch of atoms together in a lattice, I can tell you what are the energies at which the electrons can propagate freely, et cetera. But superconductivity is a qualitatively different phenomenon that's not, you know, put in by hand. It's not part of the basic equations. You have to have the imagination to ask, could something like that happen? And then the luck or the smarts to be able to figure out how to prove that it could emerge from this theoretical framework. You know, we would really like to predict unknown emergent phenomena. That is to say, derive them from fundamental law before their experimental discovery. And our record on that is not good. You know, in condensed matter physics and material physics, new phenomena, unexpected new phenomena, do get discovered on a relatively regular basis. There's things like the quantized Hall effect. There's things called topological insulators. These are all qualitatively new phenomena that follow from equations that everybody knows and believes in, but it took remarkable imagination. Well, first of all, experiment most of the time gets there first. There's a phenomenon that you don't directly understand, and you say, aha, now let me go back and see how I could derive that from the fundamental equations. Every now and then there is a triumph of imagination, and I think topological insulators qualify in that regard, but it's a pretty rare thing. So the good news is that we have so far found that our fundamental laws are able to explain all of the new so-called emergent phenomena that we stumble across as we march along studying the natural world. But it's usually after the fact. Now, I would say that life is the emperor of all emergent phenomena. There are phenomena of fundamental importance which are certainly described by the already known fundamental laws, but whose derivation from those laws completely escapes us. You know, life, surely the chief of these is life. How living matter is governed by physical principles has always been a question for theoretical physics. And, you know, despite the intrinsic difficulty of the problem, inadequate data certainly tied our hands, didn't give us anything to work with, or not enough to work with to go very far in this. Now, the ongoing explosion of quantitative biological data, like from high throughput sequencing, expression profiling, advances in microscopy, has created a totally new context for this issue. And I would say that on the biology side, it's becoming clear that we need mathematical frameworks, just like we have in physics, to extract meaning from the growing mass of data. And developing this kind of theory is what theoretical physicists do, and it's a major intellectual challenge on a par with our quest for the fundamental laws. So, now I'm getting to the heart of the matter. A quick tour of the past, present, and future of this enterprise. with a quick tour, I would try to give you a more concrete idea of what I'm talking about. Okay, so people have thought like theoretical physicists about life in the past, and there are some famous examples. I won't go through all of them. There is this well-known book, which if you haven't read, probably should read. It's a very illuminating exercise. Erwin Schrödinger, the person responsible for the Schrödinger equation for quantum mechanics, wrote something in 1944 called What is Life? And he addressed the question of what kind of thing could the unit of heredity be? What is a gene? And he made use of physical reasoning, the fact that such and such a dose of X-rays is adequate in experiments that were carried out on fruit flies to cause a mutation of a unit of life, to cause a gene to be inactivated or to change in its nature. He knew something about what is the cross-section for an X-ray to interact with an atom. He could figure out from the data on how easy it was to generate mutations in small animals from given doses of X-rays. He could make he could infer something about the size of the element that's carrying the information of heredity. And he basically concluded, after some argumentation that's well worth reading, is that a linear polymer molecule was probably the most likely thing. The size is right, the information carrying capacity is right, et cetera, et cetera, et cetera. He did not, of course, have the ability at that time to say anything more specific about it, but the line of argument is well worth reading. John Hopfield and Jacques Nignot in the 70s independently thought about the fidelity of DNA replication from the point of view of statistical physics. And they realized that there's a rather profound problem, because the energy difference between putting the right base when you're copying DNA, you have to make sure that as your polymerase runs along the single strand that you're copying, at each step you have to put in the base that is complementary to the one that is in your current reading frame. And how can you tell? Well, you can tell because the wrong base has less binding energy than the right base. That's the whole point. But the difference in energy is not very big. And if you try to estimate what would be the frequency of error just on the basis of Boltzmann statistics, energy differences, temperature, and so on, you would conclude that there would be an error rate of on the order of 1 every 10 to the 4 bases. This would be disastrous, right? It just can't be. So they inferred that there had to be a process of proofreading. In other words, it couldn't just be equilibrium statistical mechanics. There had to be some enzyme that burns energy that basically checks that you've done the right thing. And they invented schemes in which by consuming energy, you could essentially square the error rate. That is to say, take a small number and raise it to the second power. So 1 in 10 to the 4 could become 1 in 10 to the 8. And such schemes are absolutely essential for the stability of the hereditary material of life, and such things do exist. And I won't go further than that. In the future, well, there are really big theoretical questions that one would like to be able to address. The basic one is, well, what is it about non-equilibrium statistical mechanics that makes it possible for populations of distinct, stably reproducing entities to arise and become more complex over time? We hardly know where to begin in trying to answer this question, but it's a question that naturally arises if you think about starting with a chemical soup at 3.9 billion years ago and arriving at Sao Paulo after only a few billion years. As I say, we hardly know where to begin. Are there useful equations that describe the evolution of organisms with thousands of genes? Can they be solved? Can anything be said about their global behavior? People are nibbling away at that problem. Can we capture the dynamics of a cell? Can equations accurately describe its behavior, given that a cell has thousands of interacting parts? Can we see tissue types as basins of attraction of a large dynamical system? this is cellular biology. There's neurobiology. Brains carry out tasks that have an abstract representation. Can dynamical network models capture the processing power of a human brain or eye? So there are a bunch of big questions that a future theory or theoretical physics of biology will want to tackle, and nobody knows how to attack these questions. So all that one can do now is to break things down into smaller pieces and tackle small sub-problems just to convince oneself that at least small parts of the problem are doable. Okay, so what I was going to do here was to start telling you about a couple of specific things that people who are sort of motivated by the kinds of considerations that I've just gone through are working on. So I have a little menu here of things that I could tell you, and maybe what I will do is I'll just pick one and try to walk you through it as an example of what happens when people who are trained to think like theoretical physicists try to start thinking about some real biological problem in the modern context. Okay, so this is a superficial tour of what theoretical physicists are doing to attack a specific problems that fall under some big general headings. So what I'm going to do is focus on this one. Biological entities respond to stimuli, but noise limits how much they can know. Do quantitative measures of information give us new views of biological function and fitness? Okay, so what I'm going to tell you about is something that has to do with development, embryology, how a single fertilized egg can turn into a complicated adult organism. This is really quite an amazing thing, you know. We don't think about it enough, and there's, of course, a gigantic swath of biological science which essentially follows in detail what happens as you develop from a fertilized egg into an adult organism. I'm going to look at a very small swath of that problem that can be addressed in considerable quantitative detail for an annoying organism called the fruit fly. The fruit fly is it's an interesting model organism. It's been studied to fairly well. Its genetics is well understood and highly manipulatable, and you can measure all kinds of good stuff in it. And let me see if I can give you a really sort of 5-minute tour of what you can do by thinking about it. Okay, so when you try to build a fly embryo, you start with an egg, and the egg inside it has, so to say, material that will provide food and energy for the developing embryo that is going to develop inside an egg case. So this is a little bit like the egg that you eat for breakfast. This is a smaller egg. It's about 1 millimeter long. And development is a cascade of turning on of certain genetic programs. All right, so the previous slide says one of the most important phenomena in cellular biology is the ability to tell a gene to start making the protein that that gene codes for. You don't want all the genes being made all the time. You want just the right things to be made at just the right time. It turns out, and so how is this done? Well, there are proteins called transcription factors whose job it is to, when their concentration is big enough, they bind to a region just upstream of a gene, and when they do so, they enhance the rate at which that gene is transcribed. That's what a transcription factor does. And roughly speaking, the idea is the rate at which this gene is transcribed and creates, makes the protein that it is coding for is affected by what is the concentration of the transcription factor that controls that gene. So there's an input-output relationship. That is to say, let's call the concentration in the cell of this transcription factor protein, it's just a big molecule, it can have a concentration. Let's call that concentration C, and then in the cell, if we're interested in one particular gene, let's look at the output protein, the protein that this gene codes for, and let's just look at what's the concentration of the protein that's being made. The more the gene is being transcribed, the more protein should be made. So there's an input-output relation. As I increase the concentration of the guy that tells the gene to turn on, then the thing that's being made should appear in the cell at a higher concentration. And so there is a input-output relationship that you could in principle measure, right, for any one of these guys. And one of the things that's important to note is that the output for a given input is a little bit noisy, so that if you look at the output, you can't uniquely identify what's the input. So this is a not infrequent phenomenon in electronics or communication theory. You have some kind of switching device that has to decide, now I want to be on or now I want to be off, and it has to do that on the basis of some fluctuating signal. And you can characterize the noisiness of both the input and the output. Okay, so this is how gene expression is regulated. And inside the egg, there's this cascade of phenomena that occur. So you have this egg sac. Inside it, there is one fertilized, it is a fertilized egg, and it has one genome that is eventually going to be copied multiple times to turn this into a multicellular organism. And there is an output protein that basically eventually is going to determine whether I'm looking at the front of the animal or the back of the animal. And the mother has laid down inside the egg a transcription factor called bicoid, and it has a concentration which is high at one end and low at the other end. And when the machine starts going, what happens is that in the part of the embryo where this transcription factor bicoid is high enough, it turns on another gene called hunchback. When it's too low, that gene is not turned on. So that if you look at the embryo at this stage, you will see a concentration as a function of position of this transcription factor, and a concentration as a function of position of the protein that this transcription factor controls. And so this is what you see if you use some kind of fluorescent immunostaining technology. At some stage in the development, you see, aha, here's the smoothly decaying concentration of the transcription factor. Here's the rather sharply changing concentration of the gene product. This sequence, this kind of event is going to be replicated a number of times, and you're eventually going to have a patchwork of stripes, and these stripes are going to identify different regions of the insect's body. One is going to be the head, one is going to be the abdomen, one is going to be the thorax, one little patch is going to be where the wings are going to grow out of, and so on. So there's a series of binary decisions that get made that chop the real estate up into collections of cells that are going to have different adult fates. So this is it is a model of how development occurs. It's a rather simplified model, but it's what actually happens in these insects. Okay, so the levels of these proteins can be measured in tens of thousands of nuclei, and you can measure the distribution of the input-output pairs over nuclei in hundreds of eggs. And what you see is a noisy switch. So back here, where I had input versus output with error bars that indicated noise, that's exactly what you can measure, okay? Now, let me just briefly summarize what you might do with this if you were thinking about theoretical physics kinds of questions. Given two variables, so the input and the output concentration, that have a joint probability distribution, which is what we've seen. We've seen that for any given value of the input, there's a certain distribution of values of the output. So we have a joint probability distribution. Their correlation is best quantified by something called mutual information. And basically, it's measured in bits, and if you have 1 bit, then that means that if you just measure the output, that can tell you only that the input was either high or low. It's able to tell you about a binary decision or a binary classification. If you have more bits, more discrimination is possible. Now, as I say, what experimenters have done is to measure this distribution of the 2 variables, the input and the output proteins in this initial stage in the development of the fly embryo. And they can calculate then what is the mutual information between the input and the output. And they get some number. It's more than 1 bit. In fact, the data tells you that it's like 1.5 bits. So the fruit fly nucleus is more than a simple on-off switch. Okay, now here's where theoretical physics come in. We could ask how big this mutual information could be given the known input-output rule of the nucleus that we just measured over here. Okay, this is the input-output rule. This noise plot tells you about the full probability distribution, and there's an analytic argument to go through, which maybe I won't go through, which allows me to say I can find out how big this mutual information could be given the known input-output rule of the nucleus. This is a variational problem, and it has a simple solution. And I find when I do it that the maximal mutual information between the input and output in this first stage of the developmental cascade is 1.7 bits. The true mutual information is very close to optimal. In addition, when you solve this optimization problem, you can discover how transcription factor concentrations have to be arranged in order to achieve this optimum. And if you plot the data versus the prediction from this optimization problem, so the red is the optimal solution, that is to say, what fraction of the nuclei inside your egg have different levels of input and output, and you plot the prediction versus the data, you see that not only is the mutual information something that is interestingly close to the data, but actually the data is interestingly close to the optimal distribution of values. So this is an example. In other words, why the, so to say, ability to discriminate what's going on of the nuclei should lead to a mutual information that is close to maximum is not clear, but it is interesting to know that it is. And it is even more interesting to know that the conclusions that you discover from that in detail are about the way the nuclei are distributed is very close to reality. So, this is a small corner of a big problem, but dealt with in a way that I think begins to give you an idea of what you could hope to do if you were able to really get into this game of mathematical understanding of how biological systems really work. Okay, I didn't have time to tell you another story, which was about the immune system, but if I went into it, I would go far too long over, so I'm going to skip it and just ask you, why did I tell you these particular stories? Well, if I told you all of them, I would have told you about specific instances of a broader class of conceptually simple problems, conceptually similar problems spanning the tree of life and for which big data is available. So theme 1, which I did inadequately tell you about, is information transmission problems. They are ubiquitous, and data adequate for quantifying how well information is transmitted can now be collected in many systems. Higher organisms convert sensory inputs to spikes on neurons, and reconstructing a sensory input from the spike train is an information problem. And eukaryotic signaling converts external chemical signal into phosphorylation state of internal messenger proteins. You can quantify that with mass spec. So you can start doing really deep analysis of information problems in a wide range of biology. The theme that I didn't tell you about has to do with understanding probability distributions on high-dimensional data. It's very important. We'll leave that for a question if somebody wants to ask it. And these things exemplify in various ways the theoretical physics method of understanding complex phenomena through inference of a simpler underlying hidden but mathematically expressible mechanism. Okay, so back to the future. It seems blindingly obvious that biology will need some kind of general mathematical framework to organize the data flood that's in the offing. You know, the data sets that biologists generate are trivial in size compared to what astronomers or particle physicists have to deal with, but they're getting big, and they clearly don't, just by looking at them, tell you anything. You have to have some framework for extracting information. I'm personally rather skeptical that generic data mining approaches will do the job. My feeling is that finding and using simpler underlying structures will be key, but the deep problem is figuring out what complexities can be ignored in creating an adequate theory or model of a cell or a brain. And you know, we have ideas in physics that have been very powerful, coarse graining and universality and renormalization group ideas. They work very well in physics. Might they work in biology? We don't know. We need to rise above specific models for each specific biological system. I don't claim that physicists are uniquely equipped to do this, but they are naturally inclined to look for the generality we really need. And the example I discussed in the talk is just a pale imitation of the sort of analysis we'll eventually need. They're just baby steps. And the problem is a major intellectual challenge, just as hard and deep as the fundamental physics challenges we have solved in the past. And accepting the challenge, I think, will ensure the future vitality of theoretical physics, and it'll be good for biology as well, and should be fun. Okay? So with some acknowledgments to my esteemed collaborators, I will thank you for your attention and take questions if you have any. Thank you.
I forgot to mention, I'll just mention now. So there's a center in Princeton, CUNY in Princeton, that Kurt Callan is part of, Bill Bialik also, that we have a partnership with. And there's a student from here going there, I guess, later this year. And so if you're an undergraduate here and want to take part in this partnership, the way to do it is to enter into a competition in July that we have here. So it's called Jornada de Física Teórica. So it's all courses in theoretical physics, but there'll also be a course related to biology. So it's something to be aware of if you're an undergraduate attending. Okay, any questions?
So thank you for a very nice talk. So I have many questions. I'll try to restrict to two. The first is you avoided gravitation in your discussion about physics.
No.
So I was wondering whether what is the relation between, say, gravitons and dark matter, or whether that is where in the table of the standard model is gravitation.
No, I'm sorry. Okay, when I put the cartoon of the standard model on the board, I didn't include the graviton. Oh, okay, that's true. Yeah. On the other hand, you can't do cosmology without gravitation. Yeah. And you don't. Not only do you use Newtonian gravitation, but you use Einstein's gravitation in order to fully understand or fully comprehend and use the dynamics of universe expansion. Okay, so the geometry of the universe is important, and accounting for it using the rules of general relativity is important if you're going to do all of these analyses. You can get pretty far just with Newtonian dynamics. Now, so I didn't say much about dark matter because it's a fascinating problem, but it's one of these problems that there's a phenomenon and it's inside some kind of shell, and you simply can't, it's very, very hard to look inside of it. So just to summarize, why do we know there is dark matter? Well, in the first instance, we knew, and was discovered back in, I guess, the late 60s or early 70s, the so-called rotation curves of galaxies. You look at the Andromeda Nebula, it's a spiral galaxy, and there's organized rotational motion of the stars in the galaxy about the center. And not surprisingly, well, the velocity of rotation can be measured by Doppler effect phenomena. And so you find that the speed of rotation of stars varies as you go further and further out. The closer you are to the center, up to a point, the faster it rotates. It's like the planets in our solar system. And the rotational velocity is presumably not unrelated to the mass that is responsible for holding the rotating particle in its orbit. So if you try to match the observed velocities of rotation of stars in the galaxies as a function of distance from the center, if you try to match that to the mass inside the radius of the moving galaxy, it just doesn't work. There's just not enough mass. So luminous mass is just not massive enough in our galaxies to account for the speed of motion around the center of the particles, always assuming that everything is in some kind of dynamical equilibrium. And so the picture that has finally emerged that just explains so many different things is that, there is a component, so the galaxy mass is only 90% visible mass, 10% visible mass. The other 90% is just a cloud of things that are presumably particles of some kind orbiting about the center and more or less spherically distributed. And given that, the rotation curves work just fine. But the underlying assumption, well, what has to be the case is that these particles simply don't interact, or at least not in a significant way, with the stuff that we can see. So this is matter that interacts at best weakly with ordinary matter, but it's matter. And what is it? Is it a particle? Is it something else? And so people have been engaged in theoretical and experimental activities for the last 20 years to try to nail down if it's a particle, what is its mass? Can you detect it? Can I build a detector that will, it has to, in order to detect it, it has to interact a little bit with ordinary matter. And so people have begun to put limits on what its mass can be and how strongly it can interact with ordinary matter, et cetera, et cetera, et cetera, et cetera. And a clear answer to what is the dark matter has not emerged. From the cosmic microwave background radiation story, and even from the rotation curves of galaxies, quantitatively, the quantity of the mass, the net mass of the dark matter in our universe in various places, that's clearly understood, okay? Now, all right, so that, I don't know if that's the answer to the question you really wanted to ask. On the other hand, it's something I thought would be important to put on the table. Some people say, well, the dark matter story, if you just modify Newtonian gravity or Einstein gravity on large scales as opposed to short scales, you could perhaps accommodate all of the data. But that has not, to my knowledge, been a successful approach. So everybody believes that it's some kind of particle. But nobody has been able to put their hands on precisely what kind of particle it is. What is its mass? What are its interaction cross-sections and so on? But take the mic, though. Oh, well, okay, you'll get the next mic.
I'm happy. Did you want to go ahead and ask yours? I had to cut you off in the middle.
Right.
So you mentioned the emphasis or emphasized this notion of finding hidden interactions in prior work, and I'm wondering where or what do you think the limits will be if we know the players, and we even know how they interact, but we still don't know the system behavior? And do you think that's a job for physicists or other people?
Well, it's a job for scientists. The question is, what do you need in order to be a scientist? And that changes with time, all right? And I think what we're seeing is a sea change in what it means to be a scientist.
just going to poke on this a little bit more, but do you think that is something, when you think about the biological future of physics,
Yeah.
do you think that's because physicists will become less of the physicists they used to be, or other scientists will do the job that we are just exchanging about?
Well, I think physicists have a momentary competitive advantage that could readily be eroded. In other words, nobody trains biologists to do the kind of mathematical analysis except you. Very few people, right? It's coming, but if you think of the undergraduate biological curriculum, it just doesn't give you training in this kind of thing. And but it's exactly what you get trained in physics. You also get trained in, well, physics is, right? The following. You think about the following range of phenomena, and you think about it in the following mathematically based way. Well, I think my opinion is that it's become clear that thinking about another set of phenomena in this mathematically based way is actually going to be required for progress in science more largely defined. And so at the moment, people who have undergraduate training in some kinds of engineering, and physics for sure, some kinds of engineering, at least get the intellectual tools to work in this kind of science. I think we are cheating our biological students, the students who go into an explicitly biological track in the kind of training that they get, but there are efforts being made at various levels and various institutions to move the needle in the right direction. Did I go where you want? Yeah. No, but I think the real question is, what kind of tools, intellectual tools and training do you need to do science? And I think the question of what's physics or what's biology is getting a little bit, how shall I put it? They're diffusing into each other.
It's about your guess. Is it possible to model biological systems as consisting of blocks which are completely classical? Can you completely integrate out quantum mechanics or not?
Can I completely integrate out?
By the time you are in biology, is quantum effects completely integrated out?
Well, I think I know what you're asking. The great defect of biology, as far as I'm concerned, it's the thing that is really annoying about biology is that H-bar does not play a detectable role. Quantum mechanics is utterly irrelevant, except for creating the molecules whose interactions and whose detailed interaction is necessary for the biological function.
I call it integrating out.
What's that? You call that integrated out? Yeah, I think quantum mechanics gets integrated out when it comes to biology. So,
is there something like a formal proof of this, or at least reasonably rigorous proof, just to exclude this possibility of any quantum effects? I'm not sure if it's a rigorous question, though.
No, it's probably not. But okay, so obviously the kind of so the other thing, just to riff on the last two questions a little bit, everybody that's trained in theoretical physics loves beautiful equations, right? I mean, this is what mathematics is, what your first love was anyway. And this is a different kind of mathematical reasoning. It really is, it has a heavy element of data analysis of a inventive and hopefully innovative way. And there are theorems to be proven, but they're not going to involve algebraic geometry or number theory. They'll involve a rather different branch of mathematics, but there are the mathematics of things that are random. Stochastic mathematics is a very rich branch of mathematics. And I would say that's the branch of mathematics that has to be called on most heavily here. Unfortunately, there's a heavy input of computation in all of this, but we're used to this in all branches of science now. So we have to learn how to extend our human mental faculties to scales of problems that are just beyond our ability to think about. We have to have help in thinking. And who knows, maybe quantum computing will actually change the scale at which we can have a machine help us think.
Would this happen in biology? Would this happen in biology?
I have no idea. And I'll be dead before we find out. So I should have offered an opinion. I can't be contradicted.
Hi, Kurt. Thanks for this nice talking. So I've been working with molecular biology, actually with the crystal structures of proteins. And one of our biggest bottleneck is to produce the crystals. So we have to put all the proteins molecules in a single, especially form. So do you think that theoretical physics might help in this field? Have you ever had any experience with the crystal
No, but
structures?
I keep hearing that, what is it called? Cryo-electron microscopy is the answer to all prayers, right? So
Yep.
cryo-electron microscopy, you know better than I what it is, but basically you don't bother to form a crystal. You just take well, you take images of essentially random assortments of the same molecule, and then you use some kind of reconstruction methodology to infer, you have a bunch of things that have some X-ray scattering cross-section, right? And you just spread them over your target area in some random way. You know how many of them there are, and you know roughly something about their orientation distribution, and you get some diffraction pattern back, which is a total mess, right? But then you infer from the total mess. You try to infer what the single scatterer model is, and apparently this works. I mean, I'm quite impressed. I do not know in detail. I've never tried to understand how it works in detail, but I'm told that you can get not crystal structure. You can get molecular structures of really good resolution that for things that can't be crystallized. Is that a correct statement? Am I?
Yeah, in parts. Because I think that in crystals we have a higher resolution than
I would imagine.
in electron microscopy. So, actually the big bottle, the neck bottle is to produce the crystals. So all the crystallographers says that, and we are pursuing some kind of.
So the other thing that you hear is, well, okay, so crystallization requires that, the conditions be just right, that the molarity of this and that be just right. So if you can try gazillions of things simultaneously, right? You have, I don't know, a 10 million well device in which you have little tiny pots in which you use randomly varied conditions, and you ask, well, does one of them actually lead to a crystal? I mean, people, I believe, have tried this in the pharmaceutical industry, for example.
Yeah.
And I don't know how, they were talking about this 20 years ago. I haven't heard anything about it recently, so it probably means it doesn't really work all that well. The only thing I've heard that works is this cryo-electron microscopy thing.
Okay, so let's thank Kurt Callan again for his great lecture.
Thank you.
So there should be some food and drink upstairs, and for people from the school, let's say in 20 minutes, come back.
We have a 20-minute break?
Yes, people from the school will have it. Maybe half an hour. Okay, half an hour break. Right?
Okay.